My name is Samantha, and today I'll be talking about the Gram stain for my microbiology class. First off, staining in microbiology is done to help us see our specimen easier. And in this picture over here, these are the steps that have to be done before and after staining. So this first picture is smearing the specimen onto the glass. The second picture is the specimen drying. This third step here is very important. This is heat fixing the smear. Using heat on the specimen helps the stain penetrate into the specimen. And this fourth picture over here is adding the stain onto the specimen. This fifth picture is looking at it through the microscope. The Gram stain is a type of differential staining. Differential staining is a process that uses more than one stain. It helps you differentiate between classes of cells. And there are two classifications of Gram staining. There's Gram positive and Gram negative. So Gram positive cells are purple and Gram negative cells are pink. So all these little clusters of pink are negative, and all of the purple clusters are positive cells. There are 4 steps to the Gram stain technique. The first one is the crystal violet step. So crystal violet is your first stain. You add this to your heat fixed smear, and then your second step is at the application of the iodine. Iodine is added to form a crystal violet iodine complex. This is also known as the CVI complex. Iodine acts as a mordant so that the dye cannot be removed easily. And then your third step is your decolorization step. You add acetone to rinse the stain. And your fourth step is adding safranin. Safranin is your second stain. And once you add that, you can the color of Gram positive and Gram negative, it appears, and you can tell which is which. So this purple right here is Gram positive. This little pink right here is Gram negative. So Gram positive and Gram negative, their cells are different, and Gram positive cell walls contain a thick layer of peptidoglycan. This red layer, that's all peptidoglycan. And then that blue layer right there is periplasmic space. The green layer is the plasma membrane. Peptidoglycan is a polymer that consists of sugars and amino acids. It forms a kind of a mesh-like layer outside of the plasma membrane to form the cell wall. Gram positive does not have an outer membrane layer. It only has these 3 layers. Gram negative has 4 layers. It has this extra green outer membrane layer. I mean, it's not green, but the picture depicts it as green. So, Gram negative cell walls, that peptidoglycan layer is thin instead of thick, and the outer membrane also has lipids in it. So there are some errors that can occur during staining, and this happens if the sample is decolorized for too long. The peptidoglycan layer of the Gram positive cell wall can be damaged. The CVI complex is for inside of the cells, and the Gram positive cells can appear Gram negative. Alright, that's it.